In this video, we're going to learn all about chemical reactions and enzymes. First off, just a few basic definitions. We need to know our chemical reaction, reactants, and products. These are the terms we're going to use to describe what we see happening. A chemical reaction is a process. Notice it's not a thing, it's a process that changes one set of chemicals into some other set of chemicals. There's different versions of chemical reactions. We won't go into great detail with ours, but the two we're really looking at are basically going to be a breakdown, a reaction where something is split, or a synthesis reaction in which something is put together. The stuff that goes into the reaction are known as reactants. They're also known as substrates. You'll hear both of those words used when describing the first part of that reaction. And the stuff we're left with at the end, those are the products. Now, a couple quick examples of chemical reactions, stuff that we are going to see in class. One is lactose, which is the sugar found in milk, is broken up into glucose and galactose, two other sugars. The other one is hydrogen peroxide, the stuff you use to disinfect cuts and things like that. And it actually turns into water and oxygen, so you actually see it bubble when that happens. In any chemical reaction, there needs a little bit of energy to get that going, and we call that energy activation energy. It's, as you can see in the little animation there, like striking a match. You need to move the match and create a little bit of that friction and add a little bit of that to get the match burning. Once the match is burning, it's releasing a lot of energy, but you need to add a little bit of energy to get it going first. The same is true with any reaction. Now, there are substances that can decrease that amount of energy needed, and those are called catalysts. And what catalysts do is they speed up chemical reactions by requiring less energy to go in to start the reaction. For our purposes, any catalysts we're going to talk about are going to be enzymes. These are biological catalysts. They're things within living organisms that speed up chemical reactions. Now, the important stuff to know about enzymes are this. They are proteins. Okay, we talked about our different type of organic molecules. One of them is a protein. Enzymes are a great example of proteins. Secondly, they're substrate specific. What that means is a fancy way of saying that they only work with one reaction and one thing in that reaction. You can't just use them wherever they want. Third, they're reusable. We can use enzymes over and over and over and over again until they basically just get worn out. They're not destroyed in a chemical reaction, and they don't turn into something else. They're simply used as a tool to speed that reaction up. And lastly, enzymes are affected by temperature and the level of pH. So if you're out of those optimal ranges, they don't work as well. Okay? Our reactions that we saw before, for our examples, now we can see what happens when we add, or I should say, which enzymes we add to speed them up. So lactose, breaking down glucose and galactose, is sped up by lactase. Okay? Enzymes are going to always end in ASE. Okay? That indicates that it's an enzyme. Hydrogen peroxide, breaking into water and oxygen, is also sped up by an enzyme known as catalase. Now, this is just an example of what happens with hydrogen peroxide when it comes in contact with something that contains catalase. Okay, so in your blood, human blood, we have catalase. It's meant to break down hydrogen peroxide, which is otherwise toxic. And what we see happening here is some blood being dropped into a small beaker of hydrogen peroxide, and you'll notice the reaction that's taking place as this stuff foams up, becomes this large mound of essentially oxygen bubbles. Now, there are tons of different kinds of enzymes. You have thousands of them in your body that do all sorts of different jobs. The ones that we will talk about mostly are going to be digestive enzymes, which are things inside your digestive tract that help break down food. restriction enzymes, which are used to cut up pieces of DNA. We'll see this in a couple different places later on in the year. And then ligases, which are actually responsible for putting things back together, kind of like a chemical glue. And we'll see ligases a lot of times when we see restriction enzymes. Now, as an example, just to see visually how this process works, we have our diagram here. And it shows kind of some specifics about how the enzymes bond with the reactants in order to create the products. So we have our enzyme there. It's sucrase, okay, which is going to break down the sugar known as sucrose. Okay, notice that there is an active site, which is where the enzyme fits in. So the enzyme, or excuse me, where the substrate fits in. Once the enzyme and the substrate come together, we call that the enzyme-substrate complex. Big fancy word that just means they're stuck together and the enzyme's working on the substrate. Afterwards, when it's done, the enzyme releases the products and then cycles back through to get more reactants. So again, the enzyme is both substrate specific and reusable. Now, again, we talked about this earlier. Temperature and pH have a large impact on how an enzyme performs. They're designed to work at a specific temperature range and a specific pH range. So most of the enzymes in your body work best at 37 degrees Celsius, which is 98.6 degrees Fahrenheit, your normal body temperature. That's the temperature they work best in. If it gets too high, such as when you have a fever, or too low, if you're, hypothermic or something like that, they don't work as well. Same happens with the pH. Now, your pH in your body can change depending on where it is. Your blood is slightly basic, but a lot of spots in your digestive tract, such as your stomach, are slightly acidic. So it depends on what enzyme we're talking about and where the enzyme is found for its ideal pH range. Now we have here just a quick graph of what I'm talking about, and we see that there's an increase in enzyme activity going up in the temperature range until we get to about 40 degrees Celsius. After that, we see a very sharp downturn, and it actually goes all the way to 0 in terms of the enzyme activity. That is because once the activity, or once that heat gets too high, or we could say the same thing really with pH, once the pH is too extreme, it does something to the protein called denaturation. It denatures the protein. What that means is it takes the protein and the shape that the protein's in, it kind of melts it and breaks it down so that the active site that we saw before doesn't fit anymore. So it's as if we took this piece of clay that had a very specific shape and just kind of mushed it. So it doesn't have that shape anymore. And once it doesn't have that shape, it can't do its job. Now, there's a bunch of links here. If this video wasn't as helpful for you, you can follow those links and hopefully learn a little bit more about enzymes.